For all of those that are starting to see me, I'm Enrico Grisan. I'm associate professor at South Bank, working mainly on image analysis, biomedicine, biology and medicine, and adding some healthcare data. So I was to ask the audience what was their main background. I guess there are staff working on AI, machine learning, and then I see younger faces that with science, AI, and engineering. Okay, so probably so I can go fast in the introductory part, but I think it would be nice to recall some bits and pieces of what's behind it. We'll just understand then what's the impact when you're dealing with biomedical data. So the idea of multimodal learning. So the idea of integrating different views of patients or animals or diseases to have a more comprehensive understanding of what's going on. For example, understanding the different why a patient is responding to drug and another similar patient is not responding to drug with the same disease. Or why we are diagnosing a patient, two patients with the same disease. They have similar biomarkers, but they have completely different trajectories. And the question is why this is happening. So the idea of putting together a lot of data from different sources to this better understanding goes back to decades, centuries. But it has always been a matter of understanding the physics, the biology, the chemistry of the body and the patients, and trying to make sense of that. So a lot of manual work and a lot of theoretical work to put everything together. But of course, now that we're gathering more and more data, the new idea is, can we have, or can we add to our physiological knowledge or biological knowledge something that is completely data-driven? So the unknowns can be somehow taken into account or even modeled by machine learning, by learning something from the data. And the idea is, now we have a lot of sources of data. We have wearables, we have imaging, we have data, clinical records, social media, behavioral data. And the idea is, can we crunch everything together to characterize each patient in their own specificities? And then, in principle, if we can do that, then we can integrate all the data and then link them to, why this patient is different from this other, even if at first glance they are similar. So this is the all-encompassing domain where multimodal is going about. Different modalities, different sources of data, and we want to learn something, putting them together. So where this is all coming from, so probably all of you know, it all started with images. So computer vision was a big driver of machine learning in the way we are intending it now, so deep learning. This is from a paper a couple of years ago, but of course started with 2015, 2014 with the imaginary challenge. That is, can we understand what's going on in scenes or images? And in this case here, we are dividing images into the little pieces that they're composing. We have the field, we have the players, we have the ball, and we are labeling everything. So this was one big domain application. And that big domain that followed suit was natural language processing. Can we just throw data, text to the system and gather information about the text that you are presenting? And of course, this then took over the computer vision community, developing attention mechanisms, transformers, and training on large corpus of data to get the large language models we have today that are not only understanding what's in the data, but they're able to generate. And then, as things are going on in science, developing natural language processing, pour down or pour out, back in computer vision, and they go in more or less with the same technologies, being very successful. But then when you have the two big domains that are also big domains in our lives, were being trained or being managed and analyzed by large models, the idea was, can we put them together? We explain our worlds with text, but even if we are very able to understand the world with our images. And the first thing was, can we, if we have some links between what's in the text, what's in the image, and what's in the text, can we describe one with the other? And going back here, instead of saying, oh, this image has out we have 4 players, 1 ball, and 1 field, can we have a language description, a textual description that resembles very much the way we would describe what's in the image? So the idea is, could we do from the image generating text, or in principle, from text generating a corresponding representation in the image space? And the initial idea was, okay, how do we do that? We need a lot of data of paired text and images, and we need to train something that goes together. But the problem is, like with text, the same with image, is not putting just the single piece that in the text. So understand that there's a word, that there's a verb, that there is a syntactic structure in a sentence, or in the image that we have a foreground thing that represent an animal, a castle, something, and there is something else. But it's putting everything in context. So there are the relationships between words and the relationships between different parts of the image need to be consistent and need to be, well, allow me the term, smooth. And the way the natural language community solved the problem was through attention mechanisms. The attention mechanism is basically something like this. So you probably have all seen that, but basically is encoding words or pieces of sentences or important words with numbers, computers with numbers, and then just, okay, broadly speaking, loosely speaking, Scala multiplying all the numbers or the vector representing words with every other words in the same sentence or in the same piece of text. And this multiplication, this Scala product, tells you how similar, how frequently the word appear with another word. And these things are learned. So basically it is a weighted Scala product. So the weights are the things that are saying, oh, this word comes very often with these other words, or with these other words, and so on. And these are the basic mechanisms. So this is a sentence. We have all the words that have numerical representation with the vector, and then I start by multiplying the first vector with the second one, first vector with the second vector, first vector with the third, with the fourth, and so on. And you say, how is likely that these words goes with do, with not, with like, and so on. And then we go on with the second words and so on. And then through the learning from data, you can learn all the words in your language in principle, how frequently or how likely you are to go with each other. And then you can stack everything one upon the other to have more context, or to have, well, to increase the long-term relationship between words and between sentences and among sentences. The problem is, how do you train something like that? And the thing that was very successful was called self-attention. And basically what you do is have a sentence or have a text, take out part of the words, and try to understand and try to predict what's missing. It's fill in the blanks. What you've done when you were in primary school, probably, or when you're learning a new language, that you would exercise. And this is the way attention mechanism is trained, and our transformers are working. So learning to predict the missing word with a very probable, very likely output with a very likely word that make sense for the sentence and is very close to what we human would have put in the missing spaces. Okay, so I think the key thing to remember, or I feel that is important, especially when we're moving from natural language and broad computer vision to the biomedical field, is all these mechanisms are trying to understand the internal relation similarities or likelihood of two things going together. Then, when we have this thing, in principle, we say, oh, okay, we can do for language, but we are translating language in vectors or in embeddings. We can do with any other data we have. So in principle, we could put together language, videos, sound, whatever data we have pair of, and applying the same thing. You're just taking out part of the videos or part of the frames or part of the sound, and you try to predict what's missing. The problem with this approach, that was the first approach that was the first multimodal approach that was tried, is that language has a kind of constrained number of tokens, of words. Even if the way you can put them together is very large, still is limited. When you go to images or videos, things becomes much more complex in terms of possibilities of combining pixel colors, lighting, semantics, and even with acoustics. So the space you are exploring, you need to fill, and you need to understand the likelihood of being together of words with videos with sound grows exponentially. So it's very hard to train. But if you take that thing separately, so images on one side, text on the other, sound on the other, they work quite nicely. And of course, images were the first thing. This visual transformer, you divide images in patches, and then you try to predict the missing one. So exactly the same as with text. And the idea is, instead of learning, so what's the advantage of this? And what's important with respect to this? Because to train something, a multimodal system like this, you need to have a lot of data in triplets. So you need to have text, videos, or images, and sound, for example, that are paired together, because you need to understand the cross relationships. So you're losing the advantage of being able to self-train in the transformer way with masking things. The data you don't promote the data you can gather, because you need to assure that you have triplets of things. Whereas here, you go back to your initial system. You can have 3 different systems for text, natural language processing, images, and sound, or whatever data you have. You train them separately, so you can leverage all the unlabeled, unpaired data that you have. And then you need to devise a way of putting them together. And the way they're being put together is, well, putting another transformer on top of the embedding. So you learn syntax separately, and then the embedding that you learned that are representing image and text, then you learn how they go together. Or with Clip, you basically build a relationship table in which each part of the embedding in the text are linked to each part of the embedding of images, for example. And this is very used today. The problem of both of these approaches, they're very successful, they work very well, but if you start adding modalities, they don't scale very well. For example, Clip, if you have 2 modalities, you need to learn a 2-dimensional matrices of relationship. If you have 3 modalities, it becomes a cube. If you have 4 modalities, it's an hypercube in 4 dimensions. And so the space becomes quickly very, very large. The number of weights and relations that you need to learn. So good, but it's working only for a small amount of modalities. Going into the biomedical domain. So first thing, the obvious thing that was trained, and a lot of work is done and was part of the initial comment from Joshua Benji about, oh, radiologists will not be useful anymore in 10 years, was just doing the same exact thing we're doing for text and images, captioning. You have a lot of imaging data from radiology, for example, and we have very few radiologists to look at them. Can we have a system that automatically annotate them or give a textual report of what's in the images? And of course, this is very easy because all the machinery was already there. We have images, we have text, we put them together, and we just need to generate text from images. In the exact same way, we are captioning images in the natural space. And we can leverage the many, many millions of radiological data that we have for self-training, the textual language corresponding to the medical reports, and then we're just learning how to generate reliable things. So Benja's forecast didn't realize we are still kind of assessing how reliable are the outputs, but still for basic things, they're working very well. And when you're here, in principle, you can do also the opposite. And this is a paper from last year, just taking radiological report and getting back an image. And then this opens a lot of, well, questions, but also opportunities, because then you can generate images, and if they're good enough, they can use to train back the systems and making it good, and explore also fringe cases or diseases that are not very present in the population to improve how your system is able to recognize them. But this is very classical. We're just having images and text together, and we're just trying to caption one or generating one from the other. But the biomedical domain is much richer than just images and text. Because again, going back to the initial slide, the amount of data that we're gathering from each one of us, even in non-hospital settings, is massive. And this is all data that relates to your life, to your habits and behaviors, that then are linked to stratification, life expectancy, probability of being susceptible to diseases, to some diseases or others. So this is just a few of the available data, going from genomics, DNA, up to behavioral behavioral data. But also on the other side, the thing that you can get from the data is much richer than just telling or writing a report to say how you're doing. Because in principle, you can have, well, digital clinics, hospitals at home, self-monitoring, everything that has digital twins to try to see what would happen if, I don't know, you get an operation or you don't get an operation. Or if you apply, I don't know, a small device to keep your artery open, or you don't, or if you put one type or another, how would it change your outcome? How would it change your long-term survival or response to the treatment? So, and this opens a lot of questions and a lot of challenges that I will try. So I will try to give you an overview of what's in the field now and what are the challenges still open that we are trying to face. So first of all, how do you put together, the first challenge is, how do you put together data? In principle, if you're thinking about how we were fusing or the data from images and text, we're putting everything into the transformer, trying to find relationships. But what's inside this block could be a multi-tiered level in which at each tier you're trying to learn the relationships are different with different context and at the different hierarchy of your model. This is called early fusion, in which you're passing the modality, the raw modality or the embeddings of the modality in the system that is, again, layered like a deep network. And then at each layer, you are learning not only how they make the best of the embedding, but also how to cross-relate the two different modalities. And in principle, this is the best way we could exploit the relationship, the deep relationships among different types of data, because you relate them at each level of your abstraction. You try to make connections. But again, with this strategy, we are going back to the problem of the need of having pairs of things, because these are things you need to learn, and so you need to have pairs, and you cannot exploit the self-supervision approach. The other possibility is getting useful embeddings or useful representations or meaningful representations of each of the modalities separately, and then you put together these representations and try to make use of that. You're losing the hierarchical layering of the relationships, but you can exploit the huge amount of data that you can have for running self-supervision and representation learning from the different modalities. And this is the way most of the research is going now, because think about histology or genomics, it's the amount of data set where you are then paired is very limited, is in the thousands of patients. Whereas if you look at the amount of histology data or genetics data that you have unpaired, is in the millions or tens of millions. So basically, the hope or the assumption is that you train something from the tens of millions of data from histology, you get useful representation that can be used much more efficiently downstream, rather than having a smaller number of paired modalities and trying to find the relationships. So this is the first thing. And the second thing is, of course, the many ways self-supervision can be exploited and how you can combine. But the two things that are mostly used are, well, self-prediction and contrastive, and basically the idea is the same. You try to mask one. So the two ideas are, one is trying to have the embeddings or to force the embeddings to be similar when things representing similar things, and try to make them divergent where they represent different things. And you don't need to have necessarily paired things or paired couple of modalities. You just need to ensure that the outcome is kind of similar. And self-prediction usually is exactly the same thing we're using for transformer. So masking part of the image or part of one set of data or the other set of data or both, and trying to predict what's missing from the combination of the two modalities. And so this is very important for what we've been telling about the challenges. So keeping in mind, all transformers and the majority of multimodal learning today, well, when you're using self-supervised training, is trying to find out similarities. So through this dot multiplication, the scalar multiplication between embeddings, you're trying to find what's similar and what's not similar, and to align everything. And here, you're trying to estimate missing pieces from a modality, from information from the same modality and the other modality. These are the most explored domains in this case, in this scenario, in medicine. Usually two modalities. Here we have histology and transcriptomics or omics data that we have available. And you see that the usual pattern is very similar to what you've seen so far. You have your training and embedding for the histology imaging and embedding from the genetics. That could be tabular data, could be graph neural networks, or whatever you like. And then you're putting them together to predict outcome, usually, or diagnosis. So, very similar to what we already seen. Just data is kind of different. And then when you have this in mind, you can just leave your fantasy going wild. And you can put together imaging with temporal signals. In this case was spatial temporal signals, fMRI. So signals from oxygenation in different parts of the brains. Usually when you're doing a task or doing another task, with the corresponding signal that you have from electrical activation of the brain. But basically the same thing. You have one part here that is encoding the fMRI data, one part here that is encoding with some nuances, but basically it's just an encoding of the temporal signals and then putting them together to try having a better understanding of the patient. And then many different modalities, you can just pair them. In this case is histology and CT, computed tomography, to characterize tumors and stratify different types and different aggressiveness of tumors. And pathology and endoscopy, so images and videos. Exactly the same thing here. We will get back to this in a little moment about the challenges and why things are still puzzling and still not working as expected in the biomedical domain. And I think it goes back again to the way we are building and training our networks. So these are the open challenges that we still have. And I will be going through all of them in the next minutes. So our assumption so far is that we have two modalities that are telling the same thing. And this goes back to the way that were first developed. We have captioning or text and images, and we want to understand how we can build one from the other, explaining the image with text or generating images from text. And of course, they need to be similar. But in the medical domain, sometimes or many times, different modalities are giving you a different story of the patient. And if you try to compress these two stories, saying, oh, I only need the pieces that are telling the similar thing, that you're missing a lot of what's going on in the patient. Second thing is that if you want to build something that is useful in the clinic, so for real patients, clinical pathways are complicated. So you want to say, oh, I want an all-encompassing model that takes a patient, gives from his transcriptomic analysis to his higher level or organ imaging, put everything together, and you have a beautiful understanding of what's going on in a patient. But many times, patients have only pieces of data. And unfortunately, in transformers or in deep learning, we don't have the natural term, something that if you input this is just neutral, like 1 for the multiplication. So if you are missing things and you put if you put a 0 or a 1 as input for the missing part, you still get something that is non-neutral. It is impacting everything else, or it is impacting the outcome. And the other thing is that if you want to prove that the things you have learned are correct, things are expensive because you need to run trials, you need to get FDA approval, CE approval, and it's very expensive. So these are all the challenges that need to be tackled to have a system that works and that then could be deployed and is useful. So first thing is beyond biomedical fusion, before bimodal fusion. We've seen that clip is complicated, but even if you have self-attention and just having self-attention in all bits and places, that trying to do the cross mechanisms between different modalities is still complicated and doesn't scale very well. So even if you have a beautiful diagram like this and say, oh, I have 5 different modalities, still having enough data to train reliably that to understand all the respondents is hard. And it's even harder if, well, because clinicians need to gather the data and annotate the data, and then you might have patients with missing modalities. And again, how do you do? How you design a system that can tell you, oh, if I listen to modalities, I give the response I would have had if I was trained only these 3, without being derailed or distracted by the fact that I'm missing 2 inputs. And at the moment, there's no system who's dealing with that. The other thing is that what I was mentioning is that all the training models that we're using now are striving for aligning representations, for getting an embedding and finding the things that are similar and outputting them so that we can exploit the similarity to the downstream task of understanding what is the long-term outcome of the patient or the diagnosis. But what's working for captioning mightn't work for diagnosis or better for stratification of different patients with the same diagnosis. Because the information that you get from different levels of, or different levels of magnifications of a tissue, or from different functional information from a tissue, might tell you something different and even contradictory. So you might have an endoscopy, for example, in the case I'm working on, and the mucosal appearance of your intestine or your colon is normal, no inflammation, no lesions. But then, since for surveillance purposes, you always get some random biopsies here and there. You look at the biopsy and the histological slide of your tissue, and the pathologist says, oh, I've got neutrophil infiltration that is a sign of inflammation. And so how do you reconcile these 2 things? Or how do you exploit the things that the 2 modalities are telling you something different to have a better understanding of what the patient is and how you can manage or treat the patient? And the other challenge that I promised to go back to is you have multiple modalities. You train your system, you say, okay, my baseline system has this accuracy and all these performance metrics, and then I train my multimodal system and I get this other performance. And your expectations, and then it goes back a little bit also to the missing modality problem. Your expectation would be that your multimodal system would be at least as good as your best single model. In principle, you would like that. So you would like that the weakest model adds information when it can, but it not disrupt the performance of your stronger system. But it's not happening with current multimodal training in multimodal systems. Because in this case, there's a very classical approach with foundational models for the 2 streams, endoscopy and histology, and then you have a transformer on top of everything to learn correlations, self-attentions, and then you have the outcome. So, sorry, this, no, later on. I think there was a missing table, but anyway, the problem is, let's see if I have the table later. I think there was another table that was supposed to have another slide. The problem is, so what was happening is the 81% accuracy for one modality, 91% accuracy for the other modalities, combining them, 89% accuracy. Why it is happening? How can I tackle this and make the system behaving as we would like to be behaving, so that you have the most expert of the two systems to lead the way and use the information from the other only when necessary. And the other thing is that kind of surprising, and again, open questions. Putting together histology and endoscopy data, multimodal. But the score that we got from clinicians is based on histology data only. So they're looking at the histological slides and they're saying, oh, I'm seeing this amount of neutrophils infiltrating this tissue, and the score is 3 or 4, 5 or 0. So we're training a system with histological data that is trying to match histological scores. Unimodal. And you get some performance. Then you say, okay, I'm adding endoscopy information. But in principle, and so I'm adding some endoscopy information that is not carrying information about histological images, because this is where the clinical score is based on. But still, when you're doing multimodal, you are improving on the model histological data alone. And the question is why? What endoscopy is telling? Because the best we could do, since we're matching histological score, is getting the data where they were originated from and matching them. But instead, we're adding something that is in principle is unrelated, and we're getting something better. How is it possible? And this will be a question also for any time I read a paper that is trying to make long-term predictions for diseases or risk that are surprisingly based on unrelated data or very poor data. I'm questioning, am I seeing something that is real, or there's something going behind the scene? And my deep learning models are trying to exploit any possible spurious correlation that it can, and getting something resembling performance, but very brittle. Yeah, this was what I was telling you about before. So, and this goes back to this question, again, alignment and training. So if you want to learn something from cross modalities, for example, text and caption or two different images, genomics and histology, fine. And if you have pairs of things, or we are explaining with text what we're seeing in an image, it works fine. But what's happening if what we have is something like this? You have X-ray data and the post-emission tomography data. So this is telling you how much glucose is consumed in each specific region of the body. And this is telling you, well, how X-ray is absorbed by each tissue in the body. Unrated physical modality. Then I'm trying to do multimodal learning. And I'm trying to say, oh, can I predict pieces of this from this data? Or can I predict pieces of the PET data, the glucose consumption, from X-rays absorption data? And the answer is, it seems you can. And I would underline the word it seems, because my problem with this is there's no physics, there's no physiology that is justifying why you can get one from the other. Because they're completely unrelated measurements. So why do you get something? Or why would you hope to get something? And this is the problem that I have with this model and with the model that are claiming to predict your risk from 5 measurements: your body mass index, your age, your previous history of diseases, and your sex. I don't care if they have been trained on 500,000 data from UK Biobank and the German Biobank, and it seems it worked. My question is, why it is working? How it is different from just getting a very complicated correlation statistics? Because if I have just sex, body mass index, and disease history, how can I predict the age of the disease for each patient reliably? So either I throw away all the medicine that we have so far, or there's something that is not exactly right in what we are doing. And then I think the last challenge, and this is the most interesting challenge that we have, is that at the moment, all the multimodal learning that we're doing is assuming that all modalities are completely independent from each other. Whereas many times we have a hierarchical relationships among data. If you have genetics, transcriptomics, microscopy data, organ-wide imaging, body-wide biomarkers, and so on, there is a clear hierarchy and link between the two. It might be complicated, it might be unknown, but there is. And trying to find it out and exploit this is the last frontier of multimodal imaging, of multimodal learning in general, and something that is not tackled as yet. So learning how to go from here to here and back and how things are related and exploit them to represent the patient is the nice challenge. And this is a recent paper on network learning or network, and the idea is going back from what we had before, throwing all data into big machine learning system, was can we, before or with the learning system, also model the hierarchical relationships between the different scales and the different layers that the biology has in us, in the real world. And this is something that no one has done yet, and I think is going to be the next challenge. At the moment, everything that is done in this field is you model as a network, a network of relationships, the single scale, or the measurements you have from a single scale. And then you have the same thing on the other scale, and then you have a biologist telling you what's a reasonable link between measurements that you have at different levels. Mm-hmm. But you're not learning that. You're not exploiting the unknowns. The unknowns. You're only exploiting the knowns, the known knowns, so to say. But this will also be important for developing digital twins. At the moment, digital twins are working at the best digital twins, the most reliable ones, are working at organ levels, putting together structure, electrophysiology, mechanics, biomechanics, to have a reliable understanding of what's going on in each organ. But there's much more than that, and you're missing the other layers. So these are the challenges, still open, still working. But really, the most important thing is why things are working, if they seem to be working, and why they shouldn't. And this is just a question that bothers me a lot. And if you have a little, okay, no, I don't have time. If you want to have questions. Okay, thank you very much. Yes.
Just for the kind of last part, what's the role of kind of expert knowledge in this? Because you're talking about modeling data, and so where there's this kind of derived knowledge we have for these different scales, how will that fit in?
So at the moment, everything that we're building, especially at the lower scales, the closest one to biology, are driven by biological knowledge. So if you want to build a network of genes for a specific disease, we are going to the pathway banks, where there's database of, because genes can encode very different things. You say, oh, for this disease, these genes are in this pathway that is linked to this aspect, and then you put them together. So you actually very little data driven and a lot of biology driven. So this is where we are. For things that are more classical, something like this, usually the pathophysiological knowledge or the biological knowledge is introduced as constraints, usually in the loss function or in the way you're modeling interactions between modalities or between different measurements that you're inputting in your system. I know that is probably less than satisfactory in terms of what to do, but yes.
Thanks for the talk. Yeah, first question is just to hear your thoughts on how longitudinal data would be incorporated into these types of approaches, because maybe I missed it in the talk. I did see, there's the silent MRI and EEG, which are temporal signals,
but are not longitudinal data.
But not necessarily longitudinal data. And particularly with these transformer-based architectures, which are known to be or have limitations with respect to modeling to regressive data. Yeah, just what are your thoughts on how you integrate longitudinal data?
Yeah, that's a big question. So because longitudinal data is very important for clinical trials and for understanding life progression, let's say. At the moment is so the people who are looking at longitudinal data are mainly trying to reproduce survival modeling with deep models. So let's say you are data of patient or data of subjects, patients or healthy patient or healthy controls, and you have the data throughout time points. And the idea is the classical idea with survival analysis is I would like to find let's say you have a family of models that you can accept. Let's say exponential models or exponential models of linear combination of measurements, and you want to understand how each measurement affect your progression of the disease or progression in life or risk. So it was a very classical estimation. And of course, it has a lot of assumptions, a lot of constraints. So the idea is, can we replace this model with something that is much more flexible, like a network? And this is explored by, spearheaded by people in Oxford, doing survival analysis with deep learning. But it still has the same framework. So you have time points, and you would like to model a function that passes through your time points, and then you estimate risk. You can estimate the risk of each time point. So this is how things are now. Things might get interesting when you're able to do something more or closer to how physics and biology works, like exploiting differential equation. So all physical and biological systems, the way we model and the way we see it modeled are through differential equations, might be complicated, might be unknown, but this is their evolution in time. And if you can exploit those, now you can estimate with neural OD, for example, with neural ordinal differential equation. If you have enough data, you could assume that you could maybe learn complicated embedding and how these are leading the evolution in a smooth way. And then you can integrate things that you know with things that you don't know in a seamless way. But at the moment, longitudinal data are either considered as time series with all the limitations that you were mentioning by using either transformer or STM or GRU networks, something like that, or they put into the context of survival analysis.
Yeah, the other question is just sort of zeitgeist question about where foundation models get into this. So if you have a foundation model that's been trained on image data, you have another foundation model that's been trained on sleep EEG data, and you put those two foundation models together to have multimodal fusion.
This is usually what people are trying to do, what we did with our endoscopy and histology data. We got the VATB transformer trained on 5 million endoscopy images, and then we got the biomedical clip or the how's it called, the Google one, the foundation model for histology data, and we use those embeddings to then drive all the attention mechanisms downstream. But yeah, because we hope that representation is good. Yeah.
Great talk, thanks. You showed a slide quite early on when you were talking about fusion, I think, and it was showing combining, I think it was image data with omics data.
Yeah.
And so one modality with omics data and another modality with image data. But you could have split the omics data down into different omics modalities. I just wondered if you could say something about sort of best practice or how you decide when to treat something as a different modality, and when you treat everything as one modality. In that case, omics seems to be treated as one modality.
Well, it was one modality actually. It is. I wrote omics general, but it was one modality. It was RNA sequencing data. Yeah, exactly. So usually my take on that is that when you have different sources, there are different modalities. So if you have DNA sequencing is one modality, RNA sequencing another modality, and spatial transcriptomics another modality. So
you could take it to extremes and say
proteomics another one.
Imaging data captured by different machines, imaging the same area of the body, slightly different cameras, different specs, whatever, different modality. At some point, you can pull it all. It makes sense, I imagine, to put into one modality and you're learning.
So yes and no. In a sense, yes, you could, because it is an imaging of the same tissue or the same organ. So you could say I'm just adding variability in how the organ is imaged.
Yeah.
And the idea was, yes, this was done, and it is still done. You throw all the MRI, whatever their orientation, whatever, you throw all the MRIs of the brain or the heart to the system, and what you would like the system to learn from the data is that irrespective of orientation, for example, or if you're using a camera, if you're using a flash, not using a flash, night or day, if there's something in the image, you should be able to represent the same thing with the same information. What I found is that if you have slightly different views, slightly different modalities of the same thing, it is more effective to separate them than to bring them together. So on endoscopy data, the other paper submitted, but the idea is the endoscopy manufacturers, basically, it is a camera that is imaging the surface of your intestine. And the manufacturers devised a couple of methods to announce specific aspects of your tissue, for example, vessels that could be important, or scars or something like that. And some manufacturers use, it is a proper different optical imaging. So basically there was the lighting was different, and some others just use digital processing. So first thing we try to say, exactly as you're saying, same tissue, optical modality, so broadly it's the same thing. Just use everything, and the network will make sense the differences and exploit differences where they are, where it's sensible to do so. What we found out is that if instead you separate your slightly different views of the same tissue and you train separately, and then even you make a simple voting strategy and ensemble, the performance are much more robust when you change device, so manufacturer, source of the manufacturer of the endoscopy, when you change hospital settings, so you're much more consistent. So, and even when you learn with a style transfer, for example, to generate the missing modality, because maybe some hospital doesn't have the specific device to be generating the one you need, even if you're synthetically generating the missing modalities, the ensemble is still outperforming the putting everything together.
Okay, thank you.
But similarly.
I'm sorry, I'm gonna head out the door.
Yeah, sorry.
Thank you very much again.
Yeah. So, don't have any urgent questions? No, thank you very much. Very good. Very much. Questions about the
Yeah.
where do you get the permission?